Okay, so our last section for this chapter is about DNA repair that occurs during DNA synthesis. And so again, active link here that takes you to the section in the textbook. All right, so it's important to keep in mind that DNA replication is super accurate. So error rates only occur like 1 in like a billion or even less than that. Okay, but if you think about how like the scale of DNA replication, even that extremely low error rate can cause significant problems. So human genomes have about 3 billion base pairs. So if we just kind of math that out, then that means that every time one of our cells replicates, there's 6 potential errors in that new copy of that cell. And if we think that, if we know that there's 4 million cells that are being produced every second in the adult human body, you can start to see where this can be kind of a problem. We need to have some mechanisms that can catch those errors so that we're not seeing all these replications problems of our genomes as our cells continue to divide throughout our lifetimes. And then also we especially don't want them in our stem cells that go on to produce the next generation. So usually mutations, even if they, so these are the mistakes during DNA replications, usually they don't really have an effect. And when we talk about the nitty-gritty details of how proteins are formed, we'll see that there's a lot of wiggle room in those codons. Remember, we touched on those briefly once. So there's a lot of like built-in redundancy. So if we actually do see some mutations in the DNA, it might not even actually make it to what the protein's structure and function is. So usually we don't see the mutations that do get through. They usually don't have any effect, but they can be harmful. This is how we get cancer, is that there's a mutation in our those cells that say, oh, there's no like off switch, and so they just keep replicating. And then very rarely, extremely rarely, they might have, those mutations might actually be beneficial, and that's how we get a new alleles, new traits, new genes in a population, is through mutation. So we'll go through a few mechanisms. The first mechanism of where mutations might have or there are proofreading to reduce those error rates is so polymerase actually has a built-in proofreading feature where before it moves on to the next base pair, it actually checks to make sure that it forms those hydrogen bonds. So remember, G's and C's form 3 hydrogen bonds, A's and T's form 2. And so if it does, then cool. We'll just move on to the next nucleotide and just keep on processing. But if it doesn't, so if we have, you know, a G trying to pair with a T, they'll be like, no, we got the wrong thing. We go back, try it again. Okay, so it snips the, so that's one of the mechanisms. It's right there in the polymerase itself as the DNA is being copied. Another mechanism is that there are enzymes that their job is to look for and replace any mismatched bases. And so here in this strand, we see that there's a T trying to pair with a G, and we're like, bro, that's a problem. And so what these enzymes will do, these mismatch repair enzymes, is that on the old, the parental DNA strand, is sometimes we'll see that these bases have an extra methyl group. This is a CH3 group. So carbon and some hydrogens that are just kind of like hanging out. And so they'll say, okay, so this one, so they'll cruise along here and be like, all right, I know that this strand is the the original strand. This is the true copy. And so this one's a mistake, right? Because this is the real one. And so this T needs to not be a T. That's a mistake. We're going to change it to a C, and now we're good, right? Because if without this methylation, this extra carbon and some hydrogens, there'd be a 50-50 shot of pulling off the wrong one. But this methylation actually provides an extra step to ensure that these mixed match repair enzymes are getting it right. Okay. And then mechanism 3 is where we have some nuclease. Oh, so if the bases are damaged and they're not pairing up properly, we want to get rid of those. And so these enzymes, these nuclease enzymes, actually snip out that portion of the DNA where they have that damaged base pair. And then a different kind of polymerase can come back in and repair that hole using this other strand as a template, right? And then ligase comes in and just make sure everything's all nice and attached and snipped up properly, and then off we go. Okay, so where do mutations come from? So there's a couple of different ways. So these are induced mutations. So this could be a result of exposure to chemicals or ultraviolet rays from the sun or x-rays or kind of anything that's exterior that's affecting the genome of the creature in consideration. And then the spontaneous mutations are where those error rates aren't captured by any of those 3 mechanisms that we just discussed. So these are natural reactions that take place within the body that aren't caught by any of the proofreading mechanisms. So, and then we got these different kinds of mutations. So point mutations affect a single base pair. So if we have this whole series of DNA mutations, we might see just it was supposed to be a G, but now it's a T, right? So that's a point mutation where just one single base pair has been mixed up. We might see a transition substitution where either a purine is replaced with a purine, or we got a pyrimidine that's replaced with a pyrimidine. So we might have what was supposed to be a T, but now it's an A, right? So that's that's similar-ish, you know, we're probably going to be okay there. And then we have some of these other, these transversion substitutions where we have a purine that's replaced with a pyrimidine or vice versa. All right, so just remember the A's and T's are the purines and the C's and T, or yeah, I think I said all those things wrong, but you know. So in this image here, the purines are the A's and the G's, they have the double rings, and the pyrimidines are the C's and the T's with a single ring. Okay. So what do mutations do? Well, they might do nothing. And if they do nothing, then they're silent mutations. So in this case, where we, so again, we'll talk about this a little bit more when we get into actual protein synthesis, where we have a little bit of wiggle room and how these codons, these groups of 3 nucleotides, code for a certain amino acid. There's a bit of wiggle room in there. So if we have a mutation that doesn't affect what amino acids are being pulled in, then we don't care, right? Because the evolution doesn't know that there's a mistake there because we still end up with the same protein. So it doesn't matter. Insertion deletion mutations, or shortly, the short version is indels, these are the ones that can be particularly problematic. This is where we see a whole frame shift in the codons. And so here in this example, so we have an insertion deletion on top. So this is our original sequence. If we insert this G here where it's not supposed to be, then that messes up the whole rest of the codons from here on out. They're all left up. Or if we deleted a portion of that genome, then we would also see a mistake in how those codons are being read. And those are indel ones create the biggest problems. And as somebody who uses DNA sequences to figure out what species are which, these indels are a total pain in my butt because I can't now line up my DNA sequences with what's in the database. So, all right, that's indels. Oh, okay. Well, and that is everything that I have for you for this week. So when I get back, so next Tuesday on the 20, oh, it might even be March 1st when we do this. Let's see. Okay, when I get back, it will be March 1st, and we will do some practicing with DNA replication, and then we'll save the mutations for when we talk about protein synthesis. Okay, let me know if you have any questions, and I will see you guys in a week.